Hi, like Faye just said, my name is Kathryn Vanden Hoek. I am a biochemistry student at the University of Missouri, and this summer I've been working in the Taylor lab to understand mycorrhizal-associated nutrient economies. So in the basics of nutrient cycling, decomposers break down organic matter into inorganic nutrients, which are taken up by trees and then fall back down into the soil as leaf litter. This basic model brings up two limitations that I want to address today. The first is as the roots take up nutrients in the surrounding areas, it creates a nutrient depletion zone where the mature roots can't grow fast enough to reach the nutrients outside of this area. The second limitation is that in areas where decomposition rates are low, the plant can essentially be starved of nutrients. Thankfully, plants make symbiotic relationships with fungi that address both of these challenges. The plants provide the fungi with photosynthetically derived carbon, while the fungi provide the trees with nutrients that would have been otherwise inaccessible. And so this type of fungi is called mycorrhizal fungi, and there are two main types, ectomycorrhizae, or ECM, and arbuscular mycorrhizae, AM. These can be broadly classified into the miner and the scavenger. So EM evolved relatively recently from saprotrophic fungi, so it maintained the enzymes that decomposers use. AM has a very long coevolutionary history with land plants and associates with a wide variety of species. EM breaks down soil organic matter into inorganic nutrients that the plant can take up, while AM searches for inorganic nutrients with long, thin hyphae in areas that roots would not be able to access. EM is found typically in the nitrogen-limited highlats where decomposition rates are low, while AM is found in the phosphorus-limited tropics where decomposition rates are high. As far as structure, EM creates a sheath around the root tip as well as going in between the cortical cells, while AM goes inside of the cortical cells and creates arbuscules and coils. And so the mycorrhizal-associated nutrient economy, or MAIN framework, predicts that in forests dominated by AM trees, an inorganic nutrient economy will occur, while in forests dominated by ECM trees, an organic nutrient economy will occur. And that's referring to this relationship between the soil nutrients, the leaf nutrients, and the mycorrhizal type. But that brings up the question, which factor is driving this cycle? And so there are two main hypotheses. The first is that it's mycorrhizal driven. So the mycorrhizae uptake whatever their preferred nutrient type is from the soil, that enters the leaf nutrients, falls to the ground, reincorporates into the soil, and reinforces that specific nutrient type. The other hypothesis is that it's mycorrhizal associated. So the soil already contains a specific set nutrient composition. The mycorrhizae associate with plants in that area because they like that type of nutrient. Those nutrients enter the leaves, fall back down, reinforcing this specific nutrient type. So there are many questions that you can ask if you want to figure out exactly which factor is driving this cycle, but this summer I looked at these three questions. First, how do soil properties such as pH, moisture, nitrogen, carbon, and phosphorus content influence mycorrhizal colonization in plants? Two, do the soil nutrient conditions influence the leaf litter quality? And three, does species influence leaf litter quality? And so for our experimental design, we gathered samples from 5 known dual colonizing species in the Harvard forest across an AM to EM gradient, and we analyzed them for soil nutrient composition, leaf nutrient composition, and the mycorrhizal association. So the leaf and the soil samples were dried in the oven and then ground and analyzed on the discrete analyzer and the elemental analyzer for carbon, nitrogen, and phosphorus content, while the roots were stained and scored under a microscope for percent colonization of ECM, AM, and dual based on the structures that we found. So when we look at our data, we first wanted to look at how do the soil properties influence mycorrhizal colonization in plants. And so we looked at 5 different soil properties, the pH, the moisture, nitrogen content, phosphorus content, and carbon to nitrogen ratio, as well as looking across AM, ECM, and dual colonization. And the only relationship that we found that was significant was between soil nitrogen and AM colonization. When we looked at if the soil nutrient conditions directly influence the leaf litter quality, We looked at the same soil properties and then the quality of the leaf litter and the carbon to nitrogen ratio. And surprisingly to me, our data showed no significant relationship. Then when we looked at if the species influenced leaf litter quality, out of the 5 species that we looked at, the only one that showed any significant relationship was Pinus strobus. So what I have done this summer is one small part of a much larger project. And so this is going to be supplemented with a greenhouse experiment where Michael and Karina will look at the dual colonizing species Populus deltoides when provided with different soil nutrient types and mycorrhizal types in a 4x3 factorial design with 10 replicates. And so I want to take a minute to talk about how important this work will be. The nutrient composition of forest can give us insight into how they will respond to climate change. And if we better understand the effects of soil nutrients and mycorrhizal associations, we can create more accurate climate models and better understand the role that trees play in carbon sequestration. So I want to thank everyone here for coming and listening, as well as a special thanks to Faye, all of the interns, and the Taylor Lab.